As you know now, my name is Patrick Paine, and the topic I'm going to be talking about today is called the tethered secretome, a novel screening approach to identify rejuvenating peptides. So the outline of this talk is going to follow this basic stepwise list here. Introduce the aging projects in the Abu Ghut lab. The one that I'm going to talk about today and focus on is the tethered secretome. And then I'll give a little background, my aims, my results, next steps, limitations, and discussions. So one of the things I'm doing, but I won't go into too much right now, is I'm in a collaboration with a colleague who's revolutionizing mRNA delivery. He's using a specific method where he bioengineers the protein corona around the LNPs. And hopefully we're going to be able to combine that to do a new method of targeted in vivo partial reprogramming. So that's one of the projects that's happening in the Abu Ghut lab right now. Another is a collaboration with Google and Vivek Natarajan there. And this is to leverage AI's ability to nominate novel rejuvenating factors. And so that's another project that I'm working on. But as I said, the one that I'll be talking about today is the tethered secretome. And so, yeah, let's just begin. So aging is a fascinating phenomena to me, and I assume to everyone here. That's why everyone showed up. And there's this basic observation where as you increase in size, you also increase in life expectancy. But that within that, there are outliers. So shorter lived animals and longer lived animals. And they represent evolutionary derived mechanisms of longevity. One to me that's especially interesting is the immortal jellyfish. And this kind of represents an animal version of cellular reprogramming, where it undergoes reverse ontogeny all the way back to its initial stage in life and lives another full lifespan. So another interesting aging phenomena that kind of fascinates me is this regenerative potential that we have at different stages. So simple organisms have kind of a perfect capacity of regenerative potential, and that we lose that as we become more complex. And that for us, we actually have perfect regeneration prior to the embryonic fetal transition. So the information for perfect regeneration is actually contained within us, and we just have to discover it. So we get to the basic problem of aging, and people have seen this a lot, and it's just the decline in physiological function across all systems, along with an exponential growth in chronic diseases, with aging as a key risk factor. And the question emerges, well, how are you going to treat these things? And to do that, first you need to understand, at least some basic level, what aging is. And at its most general, it can be thought of as an increase in entropy over time, due to loss of repair mechanisms and loss of homeostasis. But more specifically, it's associated with these drivers of aging, or the so-called hallmarks of aging. And many of these have been documented, and their importance is more than just to describe or characterize, but also they're important for nominating or identifying new interventions. And so they allow for screening and identification of new treatments for aging. And so people have proposed and come up with many different strategies for targeting aging because of these hallmarks. And some of them you see here: heterochronic parabiosis, senolytics, caloric restriction, and then one that I have some background in, which is cellular reprogramming. And so I'm going to use that just as an example of the potential of aging interventions, where cellular reprogramming is a process where essentially you take a somatic cell, you convert it all the way back to its pluripotent state. But during that process, not only is the cell identity reversed, but also so is the cellular age. And so this represents what is actually possible now, which is true rejuvenation at the cellular level. But one thing about that specific method is it's difficult to translate. And so for my project, when I joined the Abu Ghut lab, was to think up a way, well, how can we come up with translational factors that could be more utilizable in a clinic setting, or quickly move to the clinic? And so the idea was to instead tap into the secretome. And the secretome is essentially, it consists of a subset of your total proteome. And the numbers vary, but 13 to 20% or so, consisting of growth factors, chemokines, cytokines, and et cetera. Now, the value of it is that it helps to they're essential for homeostasis. And so also it's, because they don't have to be inserted into the cell, they work on the external surface of the cell, they're easily translatable. So the next question about the secretome, well, okay, that could be useful, but is there any background on it related to aging? And so if you know anything about senescence, you know that it's an important part of the senescent-associated secretory proteins, and that senescence is beneficial at certain points in development and repair and regeneration, but it can also be harmful when persistent and detrimental. And so this is an example where secretory proteins can modify aging, although in the negative direction. And then there are also examples of secretory proteins that can modify aging in a positive way, such as GDF11, which is known to improve brain vasculature, and then Klotho, which can reduce frailty in animal models, and then also ghrelin, which can extend lifespan in progeria models. And so this kind of establishes the baseline of what the potential of a secreted protein or the secretome contains. And although people have already started to look at secreted peptides, it's not new. I'm not the first to start to look at these. In the past, they've done it in what would be called a secretome library, where it's in an arrayed setup, where they would test an individual peptide in each well. And so you can see that the numbers that they test in those libraries can be a few hundred. But the first innovation related to this project is to test it at a much larger scale. So what we did is we created a genomic library. And so this library contains over 4,400 secreted peptides, so it's almost an order of magnitude larger. And we did this by first computationally mining databases, including the Human Protein Atlas and UniProt. We also used a vendor, Instance Bio, to synthesize these fragments and then inserted them into a lentiviral backbone. But one of the key innovations here is that what allows us to do a high throughput format with this screening is that these secreted peptides would normally be secreted outside the cell. And if you do that, whoops, let me go back one. Whoops. Anyway, let me try one more. No, that was there. Yeah, anyway, so if they would normally be secreted outside the cell, but by putting them into a backbone where they retain what is known as a PDGFR alpha membrane anchor, instead of them being secreted into the media and affecting the cells around them, they're now anchored to the cell and capable of autocrine signaling. And by doing autocrine signaling, you can now do a pooled format. You can screen, you can do an NGS sequencing on those and see which ones end up being the most abundant. And they'll be specifically related to the cell type in which they're expressed. And in this way, you can do a large-scale pooled screen. The second innovation within this project is related to multiplexing. And so normally, perhaps after a pooled screen, you would go straight to NGS. But instead, what we've done is we've combined it with several of the hallmarks of aging and cell sorted specifically for the improved aging phenotype. And this way, you now have not just a screen for secreted peptides that might improve abundance or proliferation, but you also have it within the specific category of an improved aging phenotype. And then finally, the third key innovation related to this project is the development of an aging biomarker panel specific for the cell model I'm working with, which happens to be dermal papilla cells. Now, previously, groups have done biomarker panels, and the benefit to them is that any individual hallmark of aging has some variability, and sometimes, just as when you go around in life, some older people look much younger than someone their same age. The same happens with cells when you study them in a dish and you do these hallmarks of aging. So by doing multiple, what I would call a biomarker panel, you end up seeing that you combine them into one total score, and it's much more sensitive when you're trying to validate or do a screen for something that's impacting the age identity of a cell. So as I mentioned, the model that I'm working with is dermal papilla cells. So this is the orchestrator of the hair growth cycle. So it's our goal, just to sum it up, is to see if we can regrow hair in aged mice. And so that's the application for the secreted peptides. The dermal papilla cells, the strength of this model is really that it has a very clear aging phenotype, both in vitro and in vivo. So that if you find something that works, there's very little doubt about its impact. And that's one of the obvious benefits of it. So finally, the other aspect of it is that it, as I just mentioned, has a clear aging phenotype, and the hair growth cycle, for example, one of the drivers of aging in dermal papilla cells and is the DNA damage that drives hair follicle miniaturization in some cases. And also, in vitro, it has a very strong aging phenotype. So again, if we identify something that works, we'll be able to see it very clearly. Other drivers of aging related to dermal papilla cells, and I'm just bringing this up because this is part of the screen, is lipid peroxidation. And lipid peroxidation results in protein aggregation affecting organelle dysfunction. And we end up using multiple of these hallmarks of aging within our multiplex screen. And then there's also already examples where interventions have impacted dermal papilla cells and their aging phenotype and their ability to regrow hair. And one is the depletion of senescent cells and improves their capacity of hair follicle induction. And then also the depletion of senescent cells in a progeroid animal model was able to restore hair growth. So with all of this that I've shared with you so far, this leads to the hypothesis that I'm proposing, which is that the secretome harbors translational factors that can restore hair growth. And so the approach that we're using is a large-scale tethered secretome library, as I've described, and a high-throughput multiplexed overexpression screen. And the goal is to identify these translational peptides for rejuvenation. The aims of the project are to establish this biomarker panel and a molecular frailty score. It's to develop a multiplex screening platform to identify rejuvenating peptides. And finally, we'll validate and characterize these in vitro and vivo. And I'll show you the current unpublished data set that I've developed so far. And so here we are into the results. So one of the first goals was to establish the difference, if we can identify a difference between the young dermal papilla cells and the old papilla cells. Dermal papilla cells. And so we used a variety of different hallmarks of aging and readouts. And one is cell area, total cell size. And you can see if, in this case, the blue is the young dermal papilla cell, the red is the old, and then the orange is the doxo-induced senescent cell. And there's a clear difference in cell area that we observed and significant when looking at multiple replicates. We also saw a difference in nuclear size as well, and this matches publications. Finally, looking at other hallmarks of aging to establish our biomarker panel, we saw a difference in LIPR is just the short name for the lipid peroxidation dye that we're using. These readouts are all done with flow cytometry, so the cells are stained in the dish itself, harvested, and then run through flow cytometry for their fluorescence intensity. And in this case, we saw an increase in lipid peroxidation in the aged dermal papilla cells. And we also saw an increase in this marker for C-gas sting. C-gas sting is another pathway associated with aging in dermal papilla cells, and it usually recognizes cytosolic DNA, and this is associated with aging phenotypes. So again, establishing our biomarker panel, seeing that there are these differences. In some cases, we didn't see differences, and so I'm showing you in this case with ROS, we didn't see a difference. With LISO, a non-significant trend. And then with pilot data using autophagy marker, LC3B and H3K9, we still need to do replicates there, but it looks like there might be a difference. When looking at proliferation using CTG, cell titer glow, we saw a significant increase in the younger cells versus the old. So again, just firmly establishing that these cells have a strong aging difference between young and old, so that when we perform the screen, we'll be able to identify our hits. And then a clear difference in senescence as well. And that when you look at these cells under the dish, and staining with a typical SA beta-gal stain, you can see that the ones that marked in blue are the ones that are senescent. So again, the old ones show more senescence, and that the doxo-induced show more. And then you can visually see the difference in size as well. And then finally, our last proliferation-related readout was the cumulative population doubling over time, and a clear difference between young and old between passage 3 and passage 8. So with that, we were able to combine all of those molecular frailty into one single total molecular frailty score. And as I said, the value here is that if any one readout is not a robust, strong readout for us, it's okay, because we combine them all into one. We're able to get a more sensitive age-related phenotype for this specific cell type for our screen. So now we get to the screen itself, and without going into details, this is the basic kind of workflow for the screen: the thawing of the vials, the harvesting, the spinfection, puro selection, and then finally the plating and harvesting at the different time points that you see there, including day 3, 17, and day 30. And this is the full data set. So we have scrambles as control within the screen, and you can see their spread there along the based on their log fold change. You can see that all of the peptides there, they're spread there as well, and that the top 314 or so peptides also have that log fold change relative to the total. Now, when we look at the volcano plot on the right, that is from the subset that was sorted from the lipid peroxidation group that was improved. And we only ended up having a single significant hit within that group. But from other groups at different time points, we also saw other significant hits, 9 in total that I ended up testing. And so these are the log fold changes for the significant hits that were identified through the screen. And it's too early to share the names of these peptides. I'm sorry, it'll have to wait until the next talk. But I went ahead and started validating one of these peptides in particular, and the one that I'm labeling right now is H9, and this is through subcloning from the main library. And so this is done through lentiviral transduction of the cells, and then doing each one of the readouts that I just now showed you. And you can see that there's a consistent improvement across many of these, and that when totaled together into its own partial molecular frailty score, you can see there's a significant improvement relative to the scramble control and also just to the regular aged control. And then I also, because of my background in cellular reprogramming, I compared it to 2 different versions of cellular reprogramming, and this single translational peptide performed better than 2 different versions of cellular reprogramming. So then after seeing this, the benefit of doing the subcloning from the library, we then purchased the actual peptides themselves, the sequenced amino acid sequence, and applied them to the cells in vitro. And this is what the results were when testing 3 different ones. So H9 hit 10, and hit 8. And we again saw improvement across multiple of our biomarkers, suggesting potential translational aging peptides here. So an improvement in lipid peroxidation. Again, this is all with flow cytometry. Improvement in the C-gas sting pathway, and improvement in cell size. When looking at a heterochromatin marker, we also saw an improvement there, at least with H9 and H8. And then with the autophagy marker, something very similar. And that even though I don't have the images here to show you, the visually, just looking at the cells under the dish, this is matching up, because the H9 and H8 did actually look visually improved as compared to H10 or the control. When looking at DNA damage marker, this was interesting because it was almost a counterintuitive result there. We saw an increase with H9, but a stronger improvement within the senescence, both with the flow cytometry and with the XGAL readout. And again, all 3 of them seem to be having an impact on some level. So next, this is what it looks like, the actual images of the senescent cells, the control versus the 3 peptides. And what you see, just visually, you can see that the cells appear smaller. There's a little bit less senescence relative to the control, and also more cells per view. And yeah. And this is, again, after just 4 days of treatment. So this is 4 days of treatment of the cells in the dish. And that when you combine all of these for these peptides, you end up seeing a combined molecular frailty score of a relatively robust improvement, especially compared to the transduced version when we subcloned from the library. And that H9, H10, H8, all were improving our total molecular frailty score, and it's between a 20 to 30% improvement. I don't have the RNA-seq data to show you yet, but this is from, we wanted to see if, from another paper's RNA-seq data set, if they were seeing changes in the same hallmarks of aging that we were seeing in our data. And we did end up seeing some of those, including nuclear instability and chromatin remodeling. And that we also analyzed this data set. We saw one in particular gene that was increased, which is CBX5, which is related to heterochromatin increase, which is also something we saw. Again, I'll have to share the RNA-seq data as soon as I get it at the next talk. But the results summary are that we established our biomarker panel and our molecular frailty score. Using that, we then set up our multiplex aging screen, which was successful. We then validated the initial hits in vitro, and then validated them again as just a resuspended peptide. And so the next steps for this project are going to be the RNA sequencing, as I just mentioned, along with DNA methylation clock, and then finally we'll do our hair growth assays in vivo, which are just going to begin in maybe a week or 2. And so the big picture on this is that the new frontier of medicine, as people are saying up here, is that aging is a modifiable state. And so this really needs to sink in that this is a reality, that aging is a modifiable state, and that as soon as that kind of lends to the idea of what is the future of aging medicine. And one is for longevity, but the other is also for treating age-associated chronic diseases. And so in that sense, the future looks very bright. And this is my team and the lab that I'm a part of. I'm incredibly grateful to all of them. And I'd like to say thank you to the hosts and to everyone here. Thank you for listening.